All right, I'm going to introduce you to the protein reader. Basically, what we are proposing is this high throughput protein sequencing scheme with single level precision, single molecule precision. In fact, it's actually single amino acid precision. I don't have a clicker, so I'll go here. So the idea is that what we have learned in this workshop in these last couple of days is when you age, things go bad in your body. And one of the important things to diagnose or to establish the status of your aging body is have access to these biomarkers. And one of the biomarkers that could be particularly useful is have insight into the proteins, in the protein sequencing data. And currently, there is really no good method to get this information at an accurate enough level. So you have mass spec, but that gives you just broad average distributions of what the protein sequencing is. So our objective is basically reading the sequences of individual molecules at the single amino acid precision, and this would allow you, to detect these post-translational modifications in the sequence. So I give you a few steps of how this protein reader, a little bit of the conceptual scheme of the procedure. So first you have a protein, you need to functionalize the end, and basically you attach a tag with some other functional group for developing a click chemistry. You need the click chemistry because we are going to use atomic force microscopy to pull these proteins through a nanopore and use the force to identify the signature of the amino acids. So this far we have the protein, we have a tag, we have the atomic force microscopy tip, we have another tag, and what we are going to need is this substrate, which is perforated with nanopores. What we are going to do is design these pores such that the AFM, when it's pulling a protein through one of these pores, as you can see it in the picture below. What we are going to do is functionalize these tips so that one of the ends of the protein attaches to the AFM tip. Then we are going to pull this through, and we are going to design the pores such that the force that we measure from the atomic force microscope, we are going to get signatures of individual amino acids. And I will go into a little bit more detail about this. So one of the first challenges is the pore design. So you need a pore that is big enough that the proteins go through comfortably, but you cannot go too big because then you don't sense anything. So the whole method is based on this precision of the atomic force microscope to discern between different amino acids when you pull it through. And the signatures depend on many things. So anything from excluded volume interactions, so van der Waals, the amino acids have different sizes. You have electrostatics, you have many other things. So one of the key points is design the pore. This could be at a single molecule what we would be reading. So again, you have the picture of the substrate with the nanopores. You have the AFM tips. So what we are going is to pull the proteins through. And for you, those of you that are not familiar with the AFM, what you're going to get as an output is something like this, is you get the force, so the force that this actuator is measuring as a function of extension or time. And what you correlate is these peaks. So as every single amino acid goes through, you'll get a peak in force, and then a drop, a peak, a drop. So you get this signature. So this is your metric. What we are going to do is deconvolute this, identify the signature of different amino acids, and then use this method to identify the sequence of the proteins. And because you have many actuators in parallel, but you have single molecule precision, you will have one of these curves for each actuator. And then what you're going to do is just process that data in the computer. Now, one of the main problems of these sequencing methods is that, of this method initially, was that we have the single protein idea, but we need to parallelize this. We need to make it high throughput. And it seems there is previous research, this millipede invention. So there is ways of having an array of actuators, atomic force microscopy actuators. So the idea is have a microfluidic chamber where you can have a protein flux. So here a solution with highly concentrated proteins. The solution, one advantage of this technique is that we don't need to preserve structure. So we can have a salty solution, something that denatures the protein. So they basically behave like random polymers in a solution. Then we have the functionalized tag, we have the AFM. And what we do is just bring the AFM tips towards the substrate, where there is the nanopores. Then after a period of time that we need to estimate to get just, these are design variables, but after a period of time, there could be a number of proteins with the functionalized side that attaches to the functionalized tips of the AFM. And what we are going to do from there is just pull down. And we are going to measure the force, each one of these curves for each actuator. So again, this is the signature of each protein. Then the last step of this process would be to basically detach the proteins from the tips. And we are going to do this in the same microfluidic chamber, I guess, in a bottom channel, where you can just apply this solution to break the chemical bond between the protein, the functionalized protein, and the AFM tip. And then you can repeat this again. In terms of funding sources, we were thinking that the NSF Chemical Innovation Center could be, these are government sources. So this could be suitable for that. DOE Advanced Manufacturing, we believe that this single molecule, atomic precision, if anything is atomic precision, this method would be. NIH, aging research, nanotechnology, this ultimately is applied to like high throughput protein sequencing. So all the applications to that we could tackle. Is this workshop? So I think it ran out of battery, so I turned it off. Hello?
We have another one.
I can try to be loud.
Try this
All right. Hello? Yes. So what motivated this project to begin with was these biomarkers of aging. So this could be great if you could detect this misfolded protein. So the misfolded protein supposedly comes from a corrupted sequence, right? So if you could detect traces of these proteins early on, you would have basically a very advanced, very precise diagnosis method. State of the art, there are similar ideas that have been actually are used to sequence DNA. So this pour through kind of sequencing. But it cannot be done with proteins because I guess how the DNA technology works is based on these electric current readings, and that apparently is not very feasible for proteins, but it's sort of the same idea. Then we believe that this can be a really high throughput technique based on the data that is already on DNA and the technology that people have developed to put many AFM actuators together. We believe that this could be highly parallelizable, so make it high throughput in a very short time. Real world applications, we were talking earlier, somebody mentioned that we could, it's protein sequences, so the proteome of stem cells, in vitro sequencing of proteins, everything that you can think a proteome would help you, this could do it. Technology transfer opportunities, ideally, we would commercialize this device. We could do a tabletop sort of device that everybody could have in their labs. The long-term payoff is, yeah, the access to the proteome at the single molecule precision. So that's pretty cool. Then we have a like timeline here. Very roughly, in the first 5 years, what we are going to try to do is the rational design of the pore. The pore is very important because at the end, it's going to give us the force displacement signature, which is going to help us to identify the amino acids. So we are going to use computation, molecular modeling, and theory to try to rationalize the design of these pores. And at the same time, we are going to focus on doing the single pore experiment. So that will be our proof of concept. That's what we are trying to do in the first 5 years. In 10 years, the next goal is basically to make this high throughput and start commercializing the technology in a device. And hopefully start gathering real data, like individual, so personalized protein profiles at this stage. And finally, 15 years is using this data for therapeutic like goals. That would be great. And thank you to the team. And that's it. Thank you.
I have questions from our 3 judges.
Yeah, I've had
Yeah.
question. So, you always wonder, okay, what's the current state of the art, and how does this compare to that?
Mm-hmm.
Today, we can sequence DNA,
Mm-hmm.
right? And so is the problem that although we can get a DNA sequence, it's hard for us to match which protein goes to that?
Right. So this could be more catching the process downstream. So you know exactly, because the DNA basically encodes the genome,
Yes.
but then the proteins need to be expressed, and there is post-translational modifications, epigenetic effects. So there is many things that happen from the DNA to the protein. So what we do is just basically getting information at this downstream level is very useful, because it gives you direct insight into the sequence of the proteins, rather than infer through different layers from the DNA sequence how the genes are going to be expressed in the protein. So it'll give you much better information as to what are the useful biomarkers.
Seems like a lot of the interesting questions here are going to be quantitative ones about
Yes.
how many copies of a protein do you need to have high confidence in your sequence? How much of a risk of damage during this process to the amino acids resulting in inaccurate readings?
Right.
Do we know anything about that?
We don't have anything. I could try to give you an order of magnitude of things. Like we were working some numbers in terms of the high throughput, but how like the degradation of the proteins as are going through are going to depend on the size of the pore, on the functionalization. I'm not sure about the solution that we are going to use exactly to the nature of the protein. So I guess it will depend.
And just, do we have any reason to believe that this can be taken from a sample small enough to be taken from a living organism?
My guess is, yeah. So it depends. If you can really parallelize this thing, like do 1,000 activators by 1,000 activators, even if you have a low concentration solution, there will be some of them that pick at least one protein. So this is the greatness about this technique. It's like you don't need really a high concentration. As long as one activator picks one protein, you do get that signal because you have that level of precision. So you can use very dilute solutions, I think. Because in mass spectrometry, mass spec, what you do is basically break everything, right? So then you don't have any insight into this single protein that have this weird sequence. But with this, it doesn't matter, right? Like we have the reading. So we have this single molecule reading.
Yeah, I can kind of believe that there's not a lot of statistical error once you get it through the pore. Can you talk more about the unfolding, like how you denature it enough?
Right. So we were talking earlier with Tim here about a few solutions that we could use. One that came up, it was lithium.
The tricky thing here, right, is getting it to go through the pore without breaking.
Right. So these pores, this pore should be small enough to get the force signature, but it doesn't need to squeeze too hard. Like that's not something that we need, right? We don't need to make the pore so small that it's.
No, I'm not worried about the diameter of the pore. I'm worried about how do you unfold the protein without breaking.
So in a lithium bromide solution, I think it was that denatures the protein. So they should behave like random forces.
Can you tell me more about like how reliable that is for a variety of different proteins?
I don't know.
Do you know anything?
I don't know the specifics.
Like I'm wondering if like there's certain classes of proteins where that works well, what other classes that it doesn't. Proteins that tightly bind up.
Right.
That seems interesting. Like I would think that in the first 5 years, one of the things that would be the outputs would be certain categories of proteins that you can do well and others that just don't work.
Yeah. So in terms of the components of the device, one important design challenge is the solution, right? Because this is based on having the proteins denatured in terms of random calls. If there is domains, then you have, they don't go through the pore, or there's anomalies that we are not accounting for.
Any other questions from our judges?
Oh, thank you.
Great. up. Yes, Steve?
Did you consider looking at odd-shaped pores that might be able to take a crosslink protein where you've got a branch, in a sense, where you're pulling two strands through the same pore and reading each side of it independently so that you can accommodate singly branched and cross-linked protein?
Yeah, I would put that, that's an interesting perspective. I would put that in the problem of the pore design. So, as you go down into the details, this may not work for every single protein if you have this kind of architectures, but there is nothing in principle that cannot be designed to account for that.
I would ask one more thing, and since you're basically pulling different proteins down through this thing and reading them all independently, this potentially could be used to look at complex protein solutions in terms of assessing not only the modifications of them, but the relative concentrations of different.
Absolutely, yeah. We get access, that's the greatness of this. You get access at the full distribution. So that you don't get just bulk statistics. You have access to just individual molecules and the whole, everything to the one data point. Yeah, access. Yes?
You need to know the end amino protein and your attachment to it.
Yeah, so we lost our biochemist yesterday, but that didn't seem to be an issue. Apparently, many like functionalizing the end of the protein, that seemed like the easiest step.
As long as you know what it is.
Right, but so I'm telling you, I'm not a biologist. So most proteins are finished in the same functional group or very similar groups. So there is a way of doing this easily, as far as I understand.
You can do either end.
Okay.
Any other comments from our audience members? Questions? Okay, now you can sit down.